microscopy images or anything?
No.
Just give it a few more minutes and then I'll start. Oh, power. Okay. We can get started. Recording? Great. Hi everyone. I'm Nelson Lau, Director of the Genome Science Institute, and we're going to kick off the spring semester's seminar series with a change up of, typically we have academic faculty come and give talks, but you know, genome science and genomics requires using technology from various vendors, and so we have in the spring semester two vendors that will provide an overview of their genomics technologies. And today we'll be hearing from Joseph Kelly from Parse Biosciences that will tell us about their technology that's been published in the literature as split-seq. I don't think you guys call it that, but maybe you do. And then in 2 weeks, we'll have Michael Coe from Epicipher talk about cut and run sequencing. And so the point of these technology seminars is to, provide an introduction to different technologies that you guys might be considering in your research. So the first hour will be recorded on our YouTube channel and will be archived on the GSI website and will be very science and product based. And then the second hour, Joe is graciously offered to hang around for lunch. There'll be lunch in that back room there for students and trainees who might want to talk to Joe more about the technology as well as what it's like working in biotech, because we know that there's a lot of you training in an academic environment, but you may be thinking about a future career in biotech, and our institute here is supportive of that. So we thought that this would be a great way to get not only exposure to the academic realm, which you already have here, but also what's like in industry as well. And so I encourage all of you to check out the GSI seminar series website here. We list the upcoming talks. We have 2 technology talks in January and February, and then we'll go back to academics with our first GSI seminar speaker from University of Yukon of Connecticut with Rachel O'Neill. We are always looking for people to help host seminar speakers. So if you have a person in mind, please get in touch with Dr. Ala Grishak or Dr. Rachel Flynn or myself, and we can help you figure out the logistics with hosting a seminar speaker. We still have open slots in March and May that we're looking to fill. And we're encouraging students to participate as well. So if you're a student, don't feel nervous or shy about it. We can definitely help figure that out. For industry speakers too, I think some of the students have asked, can we invite industry speakers? I think industry speakers like Joe Kelly today and Michael Coe in 2 weeks is a good example of what we can do to reach out to industry. And the reason we're having these 2 particular topics is because I also want to highlight the pilot grant competition that's going to be due in April. And so, we're envisioning offering up to 6 awards of about $10,000 each for any lab that wants to try out new technology like this. You would put in an application. We would judge the applications. Obviously, we might get more applications than there are available awards, so we'll have to make some choices and decisions on which ones we select. And then if we select your application, we can then give you the money to go purchase these kits from either Epicipher or Parse Biosciences, make the libraries, and then you can get them sequenced. And we encourage you to have them sequenced here at BU with Yuri's core over here. But to generate those libraries, GSI is offering support for the best applications to try out these technologies. Okay, so and if you have other ideas of other genomics projects, you envision liking to try out, that's what the GSI is here for. We're here to help you try out new technologies. We also encourage you to do the sequencing here at BU and encourage and support your research so that you can get that pilot data for you to then use that to apply for NIH grants and keep the research rolling, okay? And so the details are here. There's also this RFA on the Genome GSI Technology Seminars. And so if you have any other ideas, please reach out to us and hope that we have a really good spring semester program here. Okay, so without further ado, I'm going to hand it over to Joe Kelly. I don't really know much about Joe other than I know that he has served other clients here at BU. I think Angie Serrano mentioned reaching out to Joe Kelly about the Parse Biosciences technology that she's interested in trying out. We heard about it in a neurodegeneration group meeting about this. And so then I reached out to Joe, and he happened to be in town, and so he's now presenting his technology. So, Joe,
thanks, Nelson. Yeah, thanks for hosting us today. We're really excited to be a part of this series that you guys have going on. I guess you'd mentioned split-seq as the technology, and yeah, so we still refer to the method as split-seq, just so that we can kind of get that out of the way. That was the technology that was developed by our co-founders at the University of Washington back starting in like 2014. You can't hear me?
Yeah, let me get you hooked up to the microphone.
Sure.
So
I can project a little bit more. I don't want to be screaming at the people in front though.
Try this. Let's see if that works. Just clip it up. There we go. Follow this. And here's the laser pointer right here.
Okay, is that better?
Yeah.
Okay. So I guess, again, thank you, Nelson, for having us here today. Nelson mentioned split-seq as a technology and as our technology. Yes, that is still the technology. It was a method developed by our co-founders at the University of Washington. So it's my pleasure to come here and talk to you about both the method as well as what we've done to commercialize it. So I'm the technical sales manager for the Northeast. As a young and growing company, we are very excited to introduce Melissa Tyndall as our newest hire at Parse. She was almost hire number 100. She was 99. She just started this past week, and she will be transitioning into the sales manager for this region. So this is her first official Parse meeting. We're excited to have her. But yeah, let's get into the company and the technology. And I'd encourage you guys, as you have questions, feel free to ask them as we're going through, so we can have a conversation versus me just up here talking at you for the next 45 minutes. So first things, we are a new company, as I mentioned. However, we are rapidly growing, and you're seeing rapid adoption within the field. Within the first year and a half, we've already had 800 labs try out and adopt our technology. You probably recognize some of the names up on that screen as well. And that rapid adoption has already led to some very impactful publications in our short history as a company. The original method was published in Science by our co-founders, and now that we've got this commercial kit, we've got a few key publications I'd like to highlight here. The first one is in Cell Reports, where we're looking at microglia within mice and how different microglia can promote lethality, or the presence or absence of microglia can promote lethality or survival within 3 different mice strains. That was done with our nuclei fixation kit as well as our WT, so you can do nuclei or cells with our technology. The next one here is out of the Mortazavi lab, where they were looking at muscle cells and doing both short read as well as long read sequencing with our technology in both nuclei and cell fixation. They've done a really nice talk, and it's up on our website. I'd recommend you guys go and look at that if you're interested in long read sequencing or application in mouse muscle models. And then this third one here is just recently came out in Nature, where they used our kits in order to look at the different OE gene mutations in Alzheimer's, or not Alzheimer's, in COVID-19, and how that impacted COVID-19 mortality rates within mice. So again, very high impact journal and high impact and topical publication that we had here. This one in particular was in cell fixation. So One of the things that people know about us really is that we're able to scale. So if you think about the history of single cell research,
The green button.
so it really started off where you were taking individual cells into individual wells and looking at one cell in a well of a plate. That was the infancy of single cell. Then 10x and other droplet-based methods came along and vastly expanded the scale. So now you're able to look at thousands to tens of thousands of cells. The Parse technology is, from the ground up, a different approach. Just like droplet is different than in plate, we're a completely different approach than droplet and these prior plate iterations, where now you're going to be able to sequence hundreds of thousands to millions of cells in a single assay. So really, we think of ourselves as that next iteration of single cell, and going to drive the science forward from here. So other than that scalability, where now you're able to do up to 1 million cells and up to 96 samples in a single assay, some other key things to realize about our technology is that there's no instrument needed. So likely what you have in your lab already is going to be sufficient for you to run your first assay with Parse. There's some flexibility built into our technology that isn't within other technologies, so that you're going to be able to fix and store your samples. You're going to be able to experiment on your schedule. You're going to be able to sequence to the depth of your interest. Then the last thing here, we'll get to this towards the end of the talk here, it's just the sensitivity of our kits. There's nothing out there that is going to have the sensitivity that you're going to get from our kits. So how do we accomplish that scalability, and what is our product line? First, we have a mini kit, which is up to 10,000 cells as the output of the kit and up to 12 samples as the input here. And this is kind of a trial scale kit, so people try it out, see if they like it. You can then scale up to our next size kit, which is our WT or whole transcriptome kit. You can do up to 100,000 cells and up to 48 samples. If you're already doing single cell work, this is likely going to be the scale that you're already working in. You're going to be able to do, if you do 10 samples out of this kit, you get 10,000 cells per sample, roughly the same throughput. But we take it a step further at Parse, and so our mega kit, so whole transcriptome mega kit, is the one where if you want to do up to 1 million cells and up to 96 samples, you'll be able to do so. What does our workflow look like? It's pretty straightforward as far as a concept goes. You come into our platform with a single cell or nuclei suspension. So if you're already doing single cell work, likely what you're doing right now is going to be good enough to come into our platform. If this is new to you, we have a team of field application scientists that will work with you to make sure that you have good protocols in place before coming into our platform. Now, this is a step where if you're coming into our assay for the first time, there's going to be some upstream optimization, even before you get Parse in your hands. So we recommend spending some time getting this optimized. Once you come into the Parse sandbox, we've got our wet lab workflow here where you start with fixation, you then do barcoding step, followed by library prep. Coming off of our library prep kit, you go onto an Illumina sequencer, so you're ready to go sequence when you're done with our kit. And then coming off of an Illumina sequencer, you come back to the Parse sandbox where we have the software also, so that you can get your data out. The important thing to know here is that the software is included in your purchase. So you buy a Parse kit, you get the software with it, no additional cost. So before we jump into the Parse platform, let's take a step back and just understand how most people are doing single cell these days. And primarily it's using a droplet-based method where you have your single cell suspension that I mentioned. It goes into a box, and through microfluidics, you're trying to capture and encapsulate your cells with barcodes, reagents in a lipid droplet, forming some emulsion using the box. From there, inside of those droplets, you lyse the cell, so the cell gets broken open, and you barcode within that droplet. Some things to be aware of here as you're going through this process. There are some cells that don't get captured in droplet because they're too big for the microfluidics. You can also run into the case where you get multiple cells captured within a single droplet, and they have to be bioinformatically removed downstream. And then another kind of watch out here is that if you're processing multiple samples, those transcriptional profiles can be changing as you're waiting for them to go through the box. So those are some of the limitations of the current model. And as we jump into Parse, we hope to address a lot of those issues with our technology. So first, similar to what you were doing before, you have that single cell suspension as your starting point. Our first step versus going into a box is to fix and permeabilize your cells. So we're locking that transcriptional profile into place. There's no droplet formation, so any kind of cell can work here. And then as you go into the next steps, because the cells are fixed and permeabilized, we're going to use those barcodes to traverse across the cell membrane or nuclear membrane and do the actual reactions within the cell itself. So there's no droplet formation with our platform. The cell is the reaction compartment. Now, this is foundational to the approach that we take, and it really allows for us to do some really fun things. So we've got you've done your fixation. That's the first step. Once you've fixed your cells, you can put them into a freezer for up to 6 months, and when you're ready to barcode, you come into our barcoding kit. So I mentioned we have those 3 different kits, 3 different throughput sizes. The solutions all have the same workflow, virtually identical. It'll be a 2.5-day process, or a 1-day process for barcoding, but 2.5 days in total for you to do your barcoding. And this is going to be the heavy lift of the talk, so you might want to take a sip of coffee before we go into this. But this is where it gets really interesting. So Nelson mentioned that split-seq approach, and that's what this is. So a split pool combinatorial barcoding. So let's imagine that you have one of our mega kits and you have 96 samples that you want to run. So you've got them all fixed, they're in the freezer, and that morning you pull your samples out, you do some cell counts to make sure that you have the right concentrations, and then you get into the barcoding portion of the workflow, where you're going to get a series of plates in our box. They're going to come plate sealed. You just pull the plate seal off. You've got all of the reagents already in there, and now you're going to start loading your sample into our plates. So the first plate that you're going to encounter here, in each well of this plate is going to be all of the reagents that you need, and in each individual well will be a well-specific barcode. On this first plate, what we're going to do is an RT step that will incorporate our first barcode as part of that. So we've taken our RNA, generated cDNA that incorporates our first barcode. This is important because this serves as our sample identifying barcode. So we've got 96 samples now. We can tell all of them apart. Also, within each well, I mentioned earlier that people started off with 1 cell per well. We have thousands of cells in each well. So that's how we're going to be able to get to that 1 million cell total output.
Is this only DT selection, or are there any fragments?
Yeah, that's a great question. So the question was, are there oligo DTs or poly DTs? And the answer is both. We prime from both the 3' end, and we use oligo DTs to random hexamers to prime across the length of the transcript. So we're getting full transcript information with our platform. Yeah, Nelson.
And is the input of cells really important? Like, can you put too many or too few cells per well?
So the input of the cells will be predetermined based upon what you want your output to be. And so we provide you with an Excel spreadsheet that you put in what your cell concentration is, so you've done your cell counts, you know how many cells you have, and you have to determine how many cells you want per sample in the output. The macros will do the calculation for you, and it'll tell you exactly how many cells to load in there. Now, the important thing, just from a conceptual standpoint here, is to recognize that you're going to have to overload here versus what you want your output to be. So in the case of a mega kit, if you want 1 million cells out in total across this entire plate, you have to load 2.85 million cells in. And the reason why you need that excess is that you're going to lose cells just because they stick to plastic, more or less. So as that plastic binds the cells, they don't all make it through to the end. But great question. So we've got our samples loaded in here. We do an RT step, so the plate just goes onto the thermal cycle. You do an RT step. You have your sample identifying barcode on every cell within each well. And so you can tell well A1 from A2 from A3, but you can't tell cell to cell apart because they all have the same barcode. And the way that we approach being able to tell cell from cell apart within a sample is through this combinatorial method, where you take all of your cells, all of your samples, they get pooled into a common vessel. So they literally go into a 50 ml tube, they get mixed up. So now you've got all of your samples, all of your cells jumbled up, and they get randomly redistributed across a second plate that will amend a second barcode through a ligation event. So now you've got two barcodes through this process. You do that a second time where you pool and then randomly redistribute across a third plate. And you end up at this point with 3 barcodes per cell. And you can start to appreciate that with each cell, even though they started off in the same well from the same sample, cells from the same well will take unique paths across these plates. And it's that unique path that will give them a unique barcode combination. So that when bioinformatically we sequence and we look for those barcode combinations, we can tell cell to cell apart. Yeah, go ahead.
How long each barcode? How many nucleotides? What is the length?
Yeah, so how long are each barcode? They're approximately 8 base pairs long each. And there are some spacers and stuff in there. We can talk about what the overall length will be, but yeah.
How many iterations of this barcoding you do?
I'm sorry, I didn't hear.
How many iterations of this in-cell barcoding you do?
How many iterations of the in-cell barcoding? So
here you have 3.
yeah, so we're going to go through a total of 4. The last barcoding step, we actually consolidate all of your cells into what we call sub-libraries, so they get consolidated into tubes. And that's because this last barcoding step is an Illumina index, and we generate what we call sub-libraries. And so in total, you do 4 barcoding events. And the number of iterations, I think this is maybe where you're going, is that through that process, the statistical likelihood that any 2 cells will follow that same path is very low, because you have 96 potential barcodes times 96 times 96 times 16 in the case of a mega kit. So 14 million potential paths that a cell coming in plate 1 could take through these 4 barcoding steps. Now in practice, we don't use all 14 million every time. It ends up being about 1 million as total output. But that has relevancy downstream when you start to consider things like doublet rates. And we'll get into that later on here. Does that answer the question?
Yeah, to see the probabilities, having, you start with 1,000 cells in one well or 10,000 cells in one well, chances are that few cells will fall in the same well in the next iteration.
It ends up being very rare in the low single digits that any 2 cells will follow the same path.
Well, you can monitor that.
Yes. So one of the things that as we kind of think about this, I like to highlight here is that in this well, if it's not making sense from a mathematical standpoint, this cartoon shows 3 different cells that these 2, the oval-shaped one and the rectangular one, you can see that they started off in the same well, because that first barcode position is a blue barcode. And those are from a different sample than this football-shaped one, because this started off with a cyan barcode in that first one. So same sample, different sample. But even though these are from the same sample, the rectangular one went blue, cyan, magenta. This one went blue, gold, magenta. So they took 2 different paths even to get back to this third plate. So when you go and sequence it, they'll have different sequences. You can tease them apart. And that continues on for that 4th barcode as well.
Does this work well for single nuclei?
Yeah, absolutely. So the only difference for us, if you're doing cells or nuclei, is that we have 2 different fixation kits. So if you want to do cells, you buy a cell fixation kit. If you want to do nuclei, you buy a nuclei fixation kit. They are agnostic as far as whether you go into any of the 3 different scale kits downstream.
Conceptually, the reason that this works is that on the average, every cell gets like 1 like one molecule of the primer, or like how have you?
Yeah, so
How
close.
does each cell become a single reactor?
Yeah, and we'll go back to this slide. So every cell will get likely hundreds to thousands of reactions each time. It's every transcript will get one reaction. So every time that you're doing that reaction, all of these barcodes are traversing across the membrane and interacting with the transcriptome within the cell. So yes, every cell will have multiple reactions going on at any given time. But mathematically, any given transcript will only have one. Yes?
From a barcode sequence point of view, are you using the same set of barcodes for each 96 plate? So the only difference is the order that they're linked to the transcript, or are you using a different set of barcodes in the first 96 and a different set in the second 96?
So they are, I don't know specifically what they are, but there are differences across the three plates as far as the barcodes and the chemistry present there. There are some linker sequences placed in here that will prevent you from amending a barcode 3 plate to a barcode 1. So let's say a cell goes through here, gets barcoded, it goes into this plate, doesn't receive a barcode randomly. You can't then barcode beyond that.
It was just that when you were explaining it a moment ago, the implication was when you were talking about football, etc., was the same color code.
Yeah.
Meaning it was the same barcode, but it isn't. It has sequences in it that define it.
Yes. Yeah, this is very simplified just so that you can kind of grasp conceptually how those barcodes are getting added and how that leads to the diversity of barcode sequences that will get you that single cell and single transcript information. So, as I mentioned, that fourth step here is our Illumina index. So each sub-library will get one Illumina index. One thing that I like to point out here is that along the way, you still have whole cells here the whole time. So if at any point you want to take a stop, do a cell count, just make sure that things are looking good, you can do that. It isn't until after we go into these sub-libraries that we do a lysis step and do that fourth barcode amendment. Once you've gone through barcoding, you go into our library prep. So barcoding takes a day, library prep takes a day and a half, and this is a very standard library prep workflow. You can make it last as long as you want to. We recommend obviously moving through quickly just so you can get your data out. One of the cool things here is that those sub-libraries I mentioned, because they have a unique Illumina index, if you're especially savvy with sequencing already, you might recognize that you can now sequence separately those different sub-libraries and get some really nice preview information about the sequencing depth that you need to get to for the rest of those sub-libraries. Yeah.
What's the mechanism, what's the technology then? There's no lysing until the last step, but you're getting the barcode into the cell. And is there a limitation on that based on what the cell type is?
So the question was, what's the technology that allows for that? And it's a permeabilization, it's kind of the combination of everything. It's the permeabilization of the cell, and that allows for you to lock your transcriptional profile, and so cross-links your transcript within the cell. So your transcripts are in place. Your enzymes and barcodes can then freely transport across the membrane and do the reaction within the cell. So that is the core technology, the enzymatic, in-cell enzymatic reactions. Now, the limitations as far as the cell types you'd ask, with our platform, you're actually going to eliminate many of the limitations that you might find with other platforms. Because there's no microfluidics present, this can be a little bit gentler on cells that might be delicate because you're transcriptionally, you're fixing them, so that fortifies them a bit. If you have particularly large cells, there's no size limitations now because it's whatever the bore of your pipette tip is, that's your size limitation. And then just because of the ability to fix, if you're getting samples in that are maybe coming in late at night, and in other approaches you've seen them degrade in quality, because you can fix in a quick like half-hour protocol, you can now work with those samples that maybe are time sensitive as well. So it really opens up a lot of things. Once you've done your library prep, you're ready to go onto a sequencer, as I mentioned. So coming off of your Illumina sequencer, you're going to get your FASTQ file. The first step for Parse is to come into our Parse pipeline. This will do all of your demultiplexing. This is run from the command line in a Linux-based setting, so you would want to make sure that you're comfortable doing that. From there, you're able to go into our Parse reports, which is going to generate an HTML report for you where you can kind of click around and look at the basic information, quality metrics, gene matrices. Or if you are more sophisticated or you prefer third-party analysis, these are just BAM files, so you can go out into Seurat, ScanPy, whatever you prefer.
Joe? Mm-hmm. So before you get to the FASTQ file, once you make the libraries, do you QC them like you would on a bioanalyzer? Like what kind of amplicon size distribution will we look for? And then how deep do you have to sequence? Get by on a next Seq 2000, or do you have to go to a NovaSeq or something?
Yeah, so you definitely want to do some QC. We have recommendations on what that are. Throughout the process, you're going to look at something like RNA integrity using a tape station. I think we also do a fluorescent measure just based upon quantity of DNA present as part of the downstream. Then how deep you sequence is going to be entirely dependent upon your particular biological question that you're asking. If you have rare expressing genes, you're likely going to want to sequence very deeply. If you're going to just have a gene, high expressed genes, you don't need to necessarily sequence as deeply. But with our platform, those sub-libraries will allow for you to pre-sequence just a subset of them so that you can understand exactly how deep you need to sequence for the rest of them. And that can inform you as far as how deep you need to sequence. The sequencer that you go on is going to be dependent likely on the size kit that you want. You're going to need, for a mega kit, 2 S4 flow cells, but with a mini kit, you can get by on like a MiSeq for some of it. So we span that full range, and it will be dependent upon what you're asking and what you're looking to do.
Okay.
Yeah.
We were talking that you use a combination of oligo-DT and random hexamers to the cDNA, but the barcodes themselves are on those oligos, or the barcodes are ligated to the cDNA?
So the barcodes get ligated to the 3' end of your cDNA that gets generated in that first
Okay, so
RT
first
step.
cDNA reaction and then ligation.
There's 2 ligation steps, and then the amendment of the Illumina.
4 ligation steps.
So the first step is cDNA generation, and the first barcode is incorporated as part of that RT step. Then we have a ligation event in the second plate, ligation event in the third plate, and then the fourth one is just the amendment of the Illumina index. So 4 barcodes, 2 ligation events, and however you want to define the Illumina index. So if you go through the Parse reports, these are the couple examples of what you would expect to see. You get your knee drop plots, which will give you your quality metrics, saturation curves. You'll also get your UMAP plots. If you're not familiar with single cell, these are some of the most common outputs from this kind of data, where you're taking your different cell populations, and based upon the transcripts expressed within those cells, you are then grouping them according to their transcriptional profiles. Now, if you're not, if single cell is new to you and you're not 100% comfortable with this yet, even if you are doing single cell, we want to make sure that you get trained with our kits. So we have, with every purchase, onboarding with a field application scientist so that you can feel comfortable going into your assay, that your samples are going to run well. You also get access to our customer support suite with a purchase where we host different videos, different white papers that will help you along the way in generating
What can go wrong?
Yeah, so what we find, things that go wrong most commonly happen upstream of our kit. So, let's see. So the single cell or nuclei suspension step is going to be critical to your success here. If you have a sample type that is not dissociated well, or you have one that's particularly persnickety and you're not able to get a good single cell suspension, or you don't have enough cells to come into the assay, that's where you're going to want to spend a lot of time. And the good news for you is that you can do a lot of that work before you even come into a Parse workflow. Yeah.
Can you use frozen tissue to make your nuclei suspension?
Mm-hmm. Yeah, so frozen tissue works fine. Typically with frozen, we're going to find that you, as you mentioned, you're going to do nuclei. The one thing that we wouldn't be able to work with is if you have previously fixed tissue. So FFPE isn't going to work with our platform. And that's because our first step is our own fixation. So to answer your question, typically upstream, if you don't have a good sample coming in, that's going to present challenges. And once you get into that, fixation, there's some optimization that is typically needed. Just like any new assay that you're running, you want to make sure that you're able to get them prepped for the downstream. Once you get into barcoding, if you've got a good fixation, typically things work pretty smoothly there.
So even with frozen tissue though, and you work out the nuclei suspension, is there another way to check whether the RNA quality is still good in that frozen tissue that's been dissociated into nuclei?
Yeah, you can do just a standard RIN score. Actually, I don't know. Yeah, well, you'd still do a RIN score for the nuclei, but that would, again, would be upstream of our kit. And our FAS would absolutely work with you. And once you get that RIN score, if it doesn't look great, or if you don't have enough cells, or if we rely a lot on image based. Just let's look at your cells. Let's look at your nuclei. Are the membranes intact? Is there blebbing? Is there a lot of debris? Let's get a good quality sample coming in, because that's really going to be the biggest impact on the quality of data that you get out.
Do you have to? Does this kit avoid ribosomal RNA swamping out transcriptome? And are there other contaminants like mitochondrial DNA that's frequently an issue in single cell sequencing?
Yeah, so we don't see much in the way of ribosomal RNA, with the exception of plant species. And we are working to put out a white paper right now around how to deplete in plants. But mammalian and otherwise, ribosomal RNA is not a contaminating issue. One of the things that we can kind of get to here is, if you guys, are you familiar with the idea of just kind of RNA contamination, ambient RNA contamination in single cell? So let me skip ahead to that. So since we're kind of talking about other, RNA that you don't want here, figured we could jump ahead here. So for those that aren't already doing single cell, with current technologies, you generate your single cell suspension just like you would with us, and you're going to damage some cells. That's just the nature of any time you're dealing with cell or nuclei suspensions. as they go through the box, as that damaged RNA leaks out, they go through the box, they get contaminated, or they get captured within that droplet. So it's part of the solution, they get encapsulated. Then when you go and lyse the cells, you're going to have both RNA, so the contaminating RNA, as well as the RNA that you're after, present in these droplets. And once you barcode there, everything gets barcoded the same. And so you're not able to tell one RNA from the other, whether it's supposed to be there or whether it's not. So we wanted to take a look at this because we frequently got this question, like, what kind of ambient RNA contamination? Did you have a question? Do you have in your platform? We didn't really have a good answer because we weren't seeing it. And so we partnered with S2 Genomics, who we designed this experiment together where we took a mouse kidney, and they have an instrument that will dissociate tissue and generate single cell suspensions off of their instrument. And so they did that with a mouse kidney. They fixed half of the cells and sent them back to us for processing. And they kept half of the cells and they put them through a Chromium and went through the standard 10x workflow. So we didn't have our hands on any of the 10x work. We didn't have hands on ours until they were already fixed. Then we went through our workflow and went through the Parse pipeline. So just trying to be as impartial as possible here. First thing that we saw was that when we took those two data sets was that we were significantly more sensitive. In this case, almost twice as sensitive as 10x with our platform. And what we noticed when we started looking at the UMAP plots was that, yeah, we had similar clustering of cells, but we also saw this population of red blood cells, which was present in the 10x data set that wasn't in ours anywhere. And that got us thinking, well, why would this be? There was a red blood cell lysis step within the singulator protocol, so there shouldn't be any red blood cells there. And what it ended up being is that ambient RNA contamination as an issue. So when we looked at our data set, none of our cells had hemoglobin content greater than 1%. You wouldn't expect there to be hemoglobin. It was mouse kidney cells. When you look at the 10x data set, 44% of the cells had hemoglobin content. So those were red blood cells that got lysed as part of the process, and their RNA was just present in the flow through of 10x, and they got encapsulated at the same time as actual kidney cells. And therefore, they got included in that data set. Now, certainly I'm sure there are ways that you can be, I don't know that S2 is the gold standard here. Maybe in your lab it's not something that you encounter that much, but in this particular instance we did see it. Now, this isn't to say that there's hemoglobin everywhere, but what you should be more concerned, this is easy to pick out, what you should be more concerned about are the other cells that are present in the kidney that you're trying to differentiate from one another, but those are getting lysed too. And that contaminating RNA is getting captured with other cells, which leads to muddy clustering of your data sets. So why isn't that an issue with us? Remember I mentioned, we are whole cells throughout the whole barcoding process. And prior to that sublibrary formation, there's a wash step. So anything that's ambient, it's likely still getting barcoded, but it's getting washed away before we do any lysis steps, and so it's gone before we have a chance to generate that final data set. Yeah.
Have you or any of your collaborators compared for the same, say, tissue, going totally nuclei versus totally cells? It seems to me that going totally nuclear extraction gets around a lot of problems. For instance, being able to get the cells out of a very fibrotic tissue, for instance.
So we don't have a data set specifically comparing nuclear versus whole cell, but that is an emerging space within the research, and RNA velocity is kind of the term. If you weren't familiar with that, genes are getting turned on and off, and so you're going to have varying levels of RNA transcript present in the nucleus versus the cytosol. And by looking at that and analyzing it, you can determine whether a cell is getting turned on or off, is the idea behind that. So I would imagine we'll see those coming out with particular labs that are interested in it. We just haven't done it. But I guess, backing up.
But from a technical point of view, nuclei, is a good medium, as it were, to be able to go through the various plates and steps as robust as they were whole cells.
Yeah. It's very much a tool in the toolbox, and I think the idea is you use the one that fits your sample best and answers your biological question. And we've done the nuclei comparison as well, if you want to take a look at that. Different setup, different tissue, where we work with mice brain. So 2 mice and third-party tissue provider cut them each in half. They flash froze, so someone had asked if frozen material works. This is an instance where we froze brain material. Half came to Parse for our workflow, and half went to a third-party 10x service provider for their workflow. So same brains, we never had our hands on the 10x, then they came back for sequencing. And similar to what we saw with the kidney, we saw significantly higher sensitivity with our platform versus 10x. And again, in the case of this second mouse brain, we saw roughly twice the sensitivity. Now, what we didn't touch on last time with that last data set was just what's the big deal with the sensitivity? Like, I'm already getting cell clustering. I'm getting all the information that I need. But where the sensitivity is going, we're going towards differentially expressed genes. So in your current data set, yeah, you're probably doing a great job of calling cell types, but within that cell type, what are the differences within their transcriptional profiles? What is causing one cell to act one way versus another within that same cell type? And that's where we see a lot of our extra reads going. So we have those core genes in transcripts that are being identified by both 10x and us. They define what the cell type is. They're high expressors, it's standard. We see very similar gene mapping with regards to that. But these lower expressors, the differentially expressed genes within a cell type, we're detecting significantly more than 10x's. So 202 genes in these GABAergic neurons versus 39 with 10x. And it was a similar story down in the microwave, 543 that we detected that 10x didn't versus their 91 that they detected that we didn't. So we think that's where the real biological insights are going to come as single cell continues to mature as a technology, and that's what we're really excited about. Excited about getting this into people's hands. Yeah, Nelson.
Do you have any genome browser coverage plots of a gene from this data set to show how those reads cover the gene between Chromium, 10x Genomics, and Parse?
When you say cover the gene, do you mean across the length of the transcript? Or
Yeah. I mean, because when you're talking about sensitivity, I know with the Chromium system, those genes, their coverage is 3' bias, because it's typically priming from the 3' end. And you mentioned you're using a combination of both random priming and poly-T.
Yeah, so that's kind of where I thought you were going with it.
Yeah.
10x only primes from the 3' end, unless you buy their 5' kit, which primes from the 5' end. So you're not going to get gene coverage information from them. With us, you do. And it's an interesting setup, and it's getting into the weeds, but you can actually tease apart with our data sets whether it primed from the 3' end or if it primed from a random hexamer. So that's fully within the data set for you to take a look at. And it's been leveraged for kind of alternative splicing type studies. But that's a great question.
You don't have any slides for that showing that?
I don't have anything to show for that, but I would recommend, if you're interested in that, taking a look at that Mortazavi study, which I mentioned earlier.
Okay.
So we've kind of hopped around here, which I like. I don't know if you guys thought that was interesting, but one thing I do want to get to before we wrap this up is this idea of doublets and how our technology really minimizes these. We had a question that alluded to this earlier, as far as what's the chance that 2 cells will acquire the same barcode with us, or AKA follow the same path. 2 cells travel the same path. And the way that we measure this, and any single cell technology measures this, is through these species mixing experiments, where you take a mouse cell line, you mix it with a human cell line, into a common tube, and then you sequence them, you barcode and sequence them. Now, you would anticipate that you're going to have all mouse transcripts on one axis, all human transcripts on the other axis. When you kind of come into the middle of the plot, that's indicating that you have both mouse and human transcripts present with the same barcode, which is an impossibility. And so that will tell you that you've got two cells that are getting the same barcode when they shouldn't. They should be excluded from the data set. So we've done this with each of our 3 kits, and across each of the 3 kits, we have, again, low single-digit collision rate. To get the true doublet rate, you actually have to double this to account for human-human and mouse-mouse. Where I like to point people is this 3.2% collision rate in the mega kit, or 6.4% true doublet rate at 1 million cells. So no other study has been published that does 1 million cells. So we're already by ourselves on this. But in other technologies, even as much as 500,000 cells in the same assay, you're looking at doublet rates somewhere in the 20%, 24% range. So roughly triple to quadruple the rate that we're at. And they're scaling exponentially. So we go linearly, they would scale exponentially. So if you scale that up to 1 million cells, you're looking at throwing away half of your sequencing reads. So think about this in the relation of your budget, about how much of your sequencing budget is going to reads that are being thrown away. With us, it's going to be 3 times lower than other technologies at minimum. So that was more or less what I know we're pretty close to time, so I'm going to give any extra time for questions that we didn't cover.
Pricing?
Yeah, pricing. With our kits, the mini kit is $4,500. That'll allow you to do up to 12 samples, up to 10,000 cells. With the WT kit, it's $9,800. So you can do up to 48 samples there, up to 100,000 cells with that. And that's going to be roughly equivalent to the scale that you might be working at right now. When you do the math there, you can save up to 50% on our kits on a per sample basis. Our mega kit is, if you're driving towards that scale, that's where you're really going to save. You can save up to 90% on a per sample basis because it costs just $16,900 for that kit.
That doesn't include fixation.
No, the fixation kits are $290 each. They will fix 4 samples each and up to 4 million cells or nuclei per sample. You buy the fixation kit that you want for the sample type of interest. And so I would say if you have a project in mind, I think we've captured most of your emails. I'll send a follow-up email to everybody that was in attendance. If you have a project in mind, let's sit down, talk about what your project looks like, and we'll get you pricing for what it will actually be.
Yeah, I mean, I gather you don't do this for people, but are there facilities around that are using Parse that you can send samples to?
Yeah, absolutely. So we are happy to partner with Course so that if you have, typically the way it works is you would fix your sample since you're going to be the expert of your sample. You can do all of that sample quality control in-house. It's fixed, you send it to the core, they take it in, and they enjoy it because now they're not getting samples at 4 o'clock in the afternoon that they need to run until 9 o'clock at night. They get them, they work on them when it's convenient for them. Now we've got the Harvard single cell core is up and running with our kits. I know we've worked with the BU core already on just a preliminary run, but I think there's probably
been
some
trained, yeah, but I'm not comfortable offering it as a service.
Yeah.
The software, as you're saying, it's included in that price in terms of access to it and online, etc. But in terms of human access, if you needed to talk to a bioinformatician, I guess that's very hard to do because the number of people out there that you're selling the kit to.
We've got a bioinformatics team that is there to support you in getting your our pipeline up and running on your cluster. Now,
Within the cost that you're...
Yeah, that's included. So we want you to be able to get data out. It doesn't help us if you're not getting data. However, we don't have like a bioinformatics team that will help you with your biological insights.
Right.
That would be on you to make sure that you're interpreting your data. Yeah.
This is optimized, of course, for mammalian cells, but what about bacteria, yeasts, and other cell types?
Sure. So there is a proof of concept publication out there that our co-founder is an author on a similar technique. It leverages the split-seq technology, and it's called the microsplit. Now, that's a proof of concept. It's not commercialized by us in any way for bacteria. If you wanted to do that, the main difference is going to be around the fixation, and that's something that we'd be able to help out from a technical guidance standpoint, but wouldn't be able to say, yeah, it's definitely going to work. But I would say that it's not just mammalian cells that we're working in. So these are just some of the sample types that we've used, our customers have used for us. So I wouldn't limit ourselves. Certainly eukaryotic is where we are well positioned. Prokaryotic is where we would have maybe some off-label usage. Everybody look for your sample and let me know if it's up there. If not, hey, we always want to add more here. This is fun for us. This is all new for a lot of people, and the more that we can support you in your research and support the research community, we absolutely want to continue to do that.
Can you use a third-party fix and permeabilization protocol? So if I'm already fixing and permeating my cells in flow, will they work with this?
So you wouldn't be able to, so the question was, can you use samples that you're already fixing and running through flow and use RKit? The answer to that is no, unless you wanted to work out a protocol that used our fixation upstream of your flow or as part of your flow workflow. It really needs to be our fixation.
Back-end population that 100,000 cells is about 5 billion reads. That's kind of quite a lot. That's why I know 6,000, right?
Yeah, so if you're looking to gauge kind of where you are on the sequencing side of things and how much sequencing you could expect, as I mentioned, it's going to range. Your mini kit, you can run 1,000 cells on a MySeq, no problem. If you're trying to do a mega kit, you're going to need 1 million cells on a mega kit, 2 S4s, and in between is where you can see that range. And this is at 20,000 reads per cell, which is a pretty good starting point. Obviously, if you need to go higher, that's going to shift the calculus there. Yeah, that's a great question. So the question is, for the mega kit, is it 1 million cells per sample? And it could be. If you look at the configuration here, with your mega kit, it's a true, or with all of our kits, it's a true range. It's 1 to 12 samples, 1 to 48 samples, it's 1 to 96 samples. So if you had a tumor that you had very rare cell types and you wanted to find all of those rare cell types and define them, you could take one tumor, dissociate it, and load 2.85 million cells from that tumor and do a mega kit on a single tumor, if that's what you wanted to do. You could do anywhere in between that too. If you had 2 tumors, you wanted to do 500,000 cells per, and you just split those 2 tumors across the plate. So anywhere within that range, and then this is the output of the kit. So you divide the output by the number of samples to get your cells per sample.
How do you do multiplexing of different samples with one single kit? Like, let's say I have 4 replicates, but I want to just use one WT kit.
So the multiplexing is built right into the kit. So if you had 4 samples or 4 replicates, and let's say you had 2 samples,
yeah,
that would be, or 2. So let's say you have a control, you have your experimental, you've got 4 replicates of each. In our platform, that would equate to 8 samples. And so if you were doing the WT, you would, in the case of a WT, you'd have 48 sample identifying barcodes. You would split those 8 samples across those 48 wells based upon what the sample loading table gives you, and you're off and running. So there's nothing extra that you need to do multiplex with us. There's no cell hashing, there's none of that.
So you just mark on the 96 well initial plate where your 8 samples are going to be, and then track the barcodes from there.
Well, you don't even do that. You plug it into the sample loading table, and it will keep track of it. You just load according to the output. So it'll say load sample 1 through 1 through 8, whatever it may be. And then on the back end, our pipeline actually plugs back into that sample loading table to do your collapsing of all of your samples into all of your wells into a single sample. So no thinking whatsoever required there. You just follow the template.
You just have to know which wells to load each of the samples in.
Yeah, you don't want to misload the wells, so that would be problematic.
Yeah.
But yeah, it's pretty much.
It tells you that.
Yeah, it's as plug and play as can be here. You don't have to keep track of anything other than probably in your initial tubes which samples where.
Okay. Are there any other questions before we wrap up this first hour?
I don't know, maybe this is difficult to say, but if a lab wanted to do this themselves, they've never done this, how easy or hard is it to do?
Yeah, I would say that the critical thing, as we talked about earlier, have a good sample coming in. That's 90% of the battle. Once you get into our barcoding, you can see this is going to be some pretty serious pipetting. Now, you're going to need to have good pipetting technique and the ability to use a multi-channel micropipette. But as long as you've got that pipetting technique and you're able to do PCR reactions, you can really handle our approach pretty easily.
You guys are very good at helping us with training and everything. Their involvement in our experiment was outstanding.
So the ultimate goal would be to offer this through the core?
I don't want to say much at this point. We did some comparison studies, which we cannot talk about yet until it's presented at HGT's this conference soon. So I was very excited about this technology.
And the cost would be much less.
Not much, but less.
It's a scalability thing. So the replicates can be done in a multiplex fashion that you couldn't previously do on the fluidics system. So it's really like if you wanted to test 16 or 32 samples, that's actually cost effective with this method as opposed to doing 32 samples on a flow method.
But if you want to do one sample